Good afternoon, everybody, and thank you so much for joining us today. My name's Josie, and I'm part of the public engagement team here at Alzheimer's Research UK, and I'm delighted to welcome you to the second event in our Lab Notes online series for 2023. I've got a few slides to quickly share with you just to introduce the event today. So here they are now. So today we are hearing from Professor Fiona Ducotterd and Professor Paul Brennan. We'll also be joined by Dr. Susan Colhass, Executive Director of Research here at Alzheimer's Research UK. So Fiona and Paul will be talking to us about the Drug Discovery Alliance and the latest research taking place at their institutions. But first, before we get started, I'll go over a little bit of housekeeping. So during the event today, you're welcome to switch on the automatic subtitles using the CC button at the bottom of your screen. They are not 100% accurate during the live event, but Zoom is getting better and better at live subtitles. However, sometimes it cannot identify some of the scientific terms our speakers might be using. The subtitles will be edited so they are correct on the event recording, and we can also send a transcript of the event as well if you need. During this event, if you would like to ask a question, you need to click the Q&A button at the bottom of the screen, which will bring up the Q&A box where you can type your question. Please do submit your questions throughout the event and we'll answer as many as we can in the Q&A session after the talks. If you are having any technical difficulties, you can also use the Q&A button to let the team know. So now we're going to ask you a few poll questions because while we can't see you due to the online format of the event today, we're really keen to know a little bit more about you. So these questions ask about your reasons for attending today. So the first one is, and also to rate your current knowledge of research. So the first one is, how would you rate your knowledge of dementia research? So is it very high, high, average, low, very low? Next is, which of these best describes your reasons for attending the event today? So perhaps you have dementia or a friend or family member has dementia. Are you a carer? Is your work related to dementia? Perhaps you don't have a personal connection, but you want to find out more. And then finally, is this your first dementia research event? So have you been to one of our Lab Notes events before? Have you been to a dementia research event in person near you? We'd love to know. So I'll just give it a couple of seconds while those answers come in, and then we will share those results with you back on the screen. So there they are. So lots of people saying they have an average knowledge of dementia research. That's great. We're here today to hopefully teach you a bit more, and hopefully you'll find out a bit more. And a real variety of people here at the event today. So lots of people with a friend or family member with dementia, and lots of you, it's your first event, but most of you have come to one before. So thanks for coming back. We're really pleased to have you back. So back to the topic of today's Lab Notes, which is the Drug Discovery Alliance and the Drug Discovery Institutes. So I'm going to give a really brief overview as Fiona and Paul will be telling us more. But first I thought I'd talk about a little bit about why does Alzheimer's Research UK exist and why do the Drug Discovery Institutes exist as well? So here are a few stats to help explain that. So nearly 1 million people in the UK have dementia. 1 in 2 of us know somebody with dementia. Dementia costs the UK economy about £25 billion a year, and 6 times more is spent on cancer research than dementia research at the moment in the UK. It's the #1 cause of death. It was the #1 cause of death in 2022, and currently there are no treatments that stop or slow the progression of dementia available in the UK. We believe that the Drug Discovery Alliance and the Drug Discovery Institutes are the biggest hope that that will change. So what are they? The Drug Discovery Alliance is made up of 3 drug discovery institutes at the universities of Oxford, Cambridge and UCL. The Drug Discovery Alliance, or sometimes we call it the DDA, bridges the gap between the discovery science taking place at these universities and institutions across the UK and those drug development experts at the pharmaceutical companies. The DDA ensures that breakthroughs by scientists go straight into the hands of drug discovery experts who have the resources and know-how to turn these discoveries into life-changing treatments for people with dementia. Translational research is the process of translating scientific discoveries about a disease into new treatments. It involves repetitive work. Scientists designing, testing, and going back to the drawing board to try and figure out how to create the best drug candidates possible. In general, the process can be divided into 4 steps. So first is choosing and validating a target. A target is a protein or a pathway that plays a role in the course of a disease. It's the aspect of biology that scientists then target with their new drugs. Researchers investigate what the exact role that the target plays is in the disease and what would happen if a drug interfered with this normal functioning. Next, they need to find a hit. Researchers need to find simple molecules that interact with the target and change the way it works. These interactions are generally weak to start with, and the hit needs to be improved by repeating the process and refining those molecules. Next, they need to develop a lead compound. So scientists then investigate how to make small changes to the molecule to improve its interaction with the target. And finally, they must test that lead compound. Once there is a drug candidate that has a strong interaction with the target, scientists must test how effective that is. They do this first in cells grown in the lab, and following these initial tests, the scientists test the drug in animals, which show characteristics of the disease. These steps in cells and animals need to be completed before moving to clinical trials for proof of safety in humans. Fiona and Paul will discuss more about the early stages of this process in more detail and probably also share some examples of their current work. You may have seen recent headlines about lecanemab, which is one of several drugs in development that work by removing a protein called amyloid from the brain. Lecanemab is the first that has been shown to slow down patients' decline in memory and thinking and improve day-to-day activities. And although the effects are modest, with the potential for some significant side effects, this nonetheless heralds a new era, the arrival of a treatment that is proven to slow down the course of Alzheimer's disease. Beyond lecanemab, there are more than 140 experimental drugs in clinical testing today, with the vast majority targeting other processes involved in dementia. We'll hear more about some of these possible avenues today as well. So, of course, today, as I've said, our speakers will be talking to us more about this process of drug discovery and how new treatments for dementia will be discovered. Once we've heard all the talks, I will return to the screen and we'll move to the Q&A session. You can submit questions at any point during the event, and we will try to answer as many as we can in the time that we have. Just to say that we can't provide clinical advice in response to questions about your individual situations. So please do get in touch with our Dementia Research info line or your GP if you have specific or personal questions. We'll be hearing from Fiona first, followed by Paul, and then Susan will join us for the Q&A section of the event. So without further ado, it's my pleasure to introduce Professor Fiona Ducotterd from the UCL Drug Discovery Institute. Over to you, Fiona.
Hi, thank you, Josie, and hi, everyone. Thank you so much for joining us today. It's my pleasure to share some of the work we're doing in the Drug Discovery Alliance at UCL and also share that discussion with my colleague Paul from Oxford University. I'm going to share some slides, if you just bear with me. I just want to make sure. Okay, so I'm going to talk about how we're making medicines at the Drug Discovery Alliance that's funded by Alzheimer's Research UK, and where some of the hard-earned funding that is raised on behalf of AR UK is really put into the future new treatments for people living with dementia. So as an introduction, I'm going to tell you a little about Josie already gave a nice introduction to the Drug Discovery Alliance. I'm going to give you a little bit more detail on that and where we are in the field of Alzheimer's research right now and why we think we have huge opportunities to really change the course of the disease in multiple patients because of our increasing understanding of the various different types of dementia and the emergence of new technologies in the field. And then Paul will then go on and give you an example of programs that we're working on, and then we'll look forward to taking your questions and having some open discussion. So the Dementia Discovery Alliance is, as Josie told you, a trilogy of institutes. I'm actually a neuroscientist by training. I did my undergraduate degree and PhD in neuroscience in the UK, and then I spent the majority of my career in the pharmaceutical industry. So really working at making new medicines for diseases. And I think the reason I was brought to the Drug Discovery Alliance is this opportunity where we have access to really new emerging ideas coming out of strong university academic labs and making the first and fast steps to get these translated into treatments for people who are waiting. I think we've all got personal stories of why dementia research is important to us, and I'm hopefully going to give you a flavour of that in my talk today. Just to note that Alzheimer's Research UK are really heavily investing in this early stage of finding new therapeutics, and our groups have really benefited from funding from AR UK to transform these ideas into reality for patients. And so we're a group of over 100 now, actually. We're growing neuroscientists working on potential new treatments in dementia across Oxford, Cambridge, and UCL. And we have a mixture of disciplines in terms of technical expertise of scientists in our team that really represent the same types of technical experts that we have in the pharmaceutical industry. So we have basic biologists, pharmacologists, and also medicinal chemists, so that we can really bring together the ideas and transform those into compounds that might act on the drug target that we are interested in looking at. And we also work really closely with clinical scientists and clinical researchers in the universities that we're in and the broader community to learn about what patients need and what the mechanistic needs are in the community to change, so that we can think about that as we translate our ideas towards new medicines for patients. We have lots of collaboration. So the way we were set up in academia was that we could then use our cutting edge biotech-like expertise that are inside the institute and work with partners with ideas or technologies that can build programs to develop that into new technologies. And we've worked on over 20 targets so far across Alzheimer's, Parkinson's, Huntington's disease, ALS, and mixed dementia. So we're really interested in neurodegeneration across a lot of different diseases. We've also had the opportunity to look for additional funding to add to the core funding. Making drugs, as I'm going to tell you on the next slide, is an extremely expensive and long process. And it's really important that we have the resources to make these ideas into a reality. And at UCL, we also have spun out a company that is focused on, from one of the ideas that came out of the research in the lab at UCL and in our broader collaborations to really make these ideas and progress them towards clinical trials. And we have projects across global organizations, and we work very closely within the 3 institutes as well, which is a really rich environment for us to work in as neuroscientists and drug discovery experts. So Alzheimer's Research UK, as I'm sure a lot of you know, invests in many strategic areas, and I thought it would be nice to show you where we fit in the Drug Discovery Alliance to that real investment across the stages that Alzheimer's Research UK is supporting in terms of development. And so that starts with ideas. I think Josie gave you a nice insight into this, that we start with a target, or where we think making a chemical that modulates that target would be helpful and transformative in terms of disease biology. And then we go into looking for molecules that modulate that, and then we look for compounds and then refine those in the iterative process. And then they go into efficacy studies where we can show if they do what we hoped they would do when we modulate that target, and then go into clinical trials. And this takes a really long time. And the academic groups are really doing a lot of the cutting edge innovation in terms of identifying the targets that might be of interest in terms of a therapeutic opportunity for Alzheimer's and other dementias, and then validating those in terms of confirming what type of modulation we would need to make a drug. And then our group comes in to build screens to look through hundreds of thousands of chemical libraries to find compounds that might bind to those targets, and then we design them very carefully to make sure they have properties that would make them a drug, which means that they could be they have the right, they get to the brain, number one, and then also that they're safe and don't have off-target effects that would make them not progress into a safe and effective therapeutic. And then we also want to show that they work in the way that we expected before we go into testing in humans and confirming that disease mechanism. Our goal in the DDI is to get compounds through to that proof of concept stage, and then we would look for partners with clinical expertise to progress those drugs into humans. So in terms of the general landscape of drug discovery and development, what's our strategy at the DDA? So we always need to identify the right drug target. then we need to confirm confirm how to modulate that target in the disease to have a clinical benefit. And we do that by designing a series of experiments to find the chemicals that hit that target, modulate them, show the disease relevant therapeutic effects, and then reach the site where they're safe. And this on the right is actually from a publication, so a scientific publication that was done by AstraZeneca, which is a big pharmaceutical company, where they evaluated the success of getting an idea from the discovery stage, which we work in at the DDA, right through to the commercial success and the approval of that drug. And what they found were these 5 R's that make the best possible statistical probability of success for getting a drug from that idea to patients. And this in the corner is actually a picture of my grandmother who had Alzheimer's disease, and I was just starting my career at that time when we were watching her change, and it's really exciting for me to have the opportunity to work. And when I was watching her decline, I was constantly thinking about my lab work and how what was happening to her was happening in the lab. And I think that that's something that really inspired me to continue this work. It's my pleasure to be leading this group to really try and make transformative patient solutions for patients and make sure that this doesn't happen to other families in the future, which I think everyone in this call is committed to. So our mission in the DDA is really to identify the right novel targets for different dementias, and different dementias is going to take me on to the next part of my discussion about the complexity of dementia and the complexity of different forms of dementia, and how we are really trying to understand those mechanisms that are driving different types of dementia and the different approaches therapeutically that we might take to intervene with the different types that we are starting to understand in the field. First, I'm going to tell you, I already told you this is a very long journey, and this slide shows you really how long it takes to get from that basic research idea to a post-marketing approval. So basic research, just to understand the target, this is industry averages that came from a report done in the Japan Pharmaceutical Guide, takes from 2 to 3 years. So that's what and then the non-clinical studies, so once we know we have the right target, try making the chemical modalities that modulating it and testing them, takes another 3 to 5 years. And then when we get to the clinic, we have another 3 to 7 years, and then an NDA that takes about a year, which is a new drug application to regulators to show that the drug does what we think it was meant to in the clinical trials. And then we have to go through approval and launch. So this is a really long process. And in that phase, we are always thinking about how do we get to point 6? So even when we're in this very early research phase, we're using all the information available in the field to design studies, understand how our target might modify the disease to get that successful post-marketing to that post-marketing stage where people actually have access to the medicines. Clinical studies, just because this will come up in when I talk about the field in terms of dementia, there are 3 phases of clinical trials. The first is phase 1. That's where we test molecules, usually in healthy volunteers, just to show that we have safety and they get to the right places in the body and they do the things that we are expecting in the early stages of clinical trials. Phase 2 is usually done in a patient population to start to build understanding of the safe dosage and the administration method in a small number of patients, and to make sure that we're starting to get a confirmation of efficacy, that we are getting some understanding that these compounds are effective as well as safe in patients. And phase 3 is the big study or multiple big studies that shows that these drugs really work in patients. And we, as I said already, as research scientists, we are not working in that late stage of the development process. We're really focused in this very early stage, from basic research to non-clinical studies, and how we get those hypotheses confirmed and the molecules that modulate the hypothesis that we know we need to address, and get those compounds into patients. And just as a flag, I think it's important, just for the field to understand why this is such a long process, is these are very complex diseases. But the hypothesis for Leqembi's mechanism of action was proposed 30 years ago, and we've just seen, the marvelous new data that shows that there is clinical effects with that compound. So I think that what our goal is now is to try and speed that process up and get using the technological developments and across basic research translation and clinical research, we can facilitate that shortening. I think that I'm not going to go in so much into biomarker discovery today, but that's where we can measure disease relevant changes in patients over time using technologies like CSF samples, blood samples, and also clinical imaging, where we can do live brain structures in living patients. They're allowing us now to understand the disease course in living people in a way that in the past we could only really see once people were donating their tissue for post-mortem. So I think this is our time. It's a really exciting time to be in dementia research, and I hope that we share those feelings with you today. And we're looking forward to telling you more about that later in the discussion. So I'm going to flick through this because in the interest of time, I want to make sure we have enough time for your questions. I already told you we're looking for the right targets. We're going to validate those targets. And then we also build appropriate strategic collaborations to bring in the right expertise to really drive projects forward as quickly as possible toward patients. And we have, I think Josie showed a slide earlier about the target validation. So how do we know that we have changes in the expression of a target, for example, in human brain tissue using post-mortem staining? Then we can build screens that show that are specific to that target, where we can look for compounds that modulate it. And then we have chemists that design the structures. Paul, actually, who's on the call today, is a chemist, so he might give you some more information on how we do the chemical design for programs. And then we also run functional studies to show that the compounds in preclinical models do what we hope they will do in the clinic later. So there's multiple mechanisms of Alzheimer's, and I think and dementias. Dementias are highly complex disorders. They likely start to evolve in people way before we start to see clinical symptoms. And so it's very important that we understand the early changes that drive the disease and then look for ways of changing the course of the disease by intervening early. The brain has many cell types, the neurons, which are the most prominently discussed and are, obviously important for all of our neural processes, but there's also maintenance cells called glia, which include microglia and astrocytes, that also are very important and emerging in terms of therapeutic opportunities to modulate neuronal function by some of the support cells. We're also investigating other areas like vascular contributions, so how the brain gets its energy and the way that the blood flow can change in disease. And there's lots of different mechanisms that we're exploring using scientific and evidence-based approaches to really look for clinical benefit in the future. I think that there's a lot already in clinical development in terms of Alzheimer's disease and dementia, and this is a study that was published by a prominent researcher, Jeffrey Cummings, about the mechanisms of action for drugs that are in clinical development right now. So we're, as I already mentioned earlier, we're right way before this. So we're looking at the next generation of targets. But I think this landscape just shows you how much hope for the future there is in this space. So we're looking at lots of different mechanisms that have supportive biology behind them. Paul is going to take you through one of these and tell you about how we're looking at that in the early stages. But there's more than 126 agents in clinical trials for Alzheimer's. Most of these are in the trial, the target for disease modification. Until the newer therapeutics like lecanemab, we were really looking at only symptomatic treatment, so not treatments that could change the course of the disease. So we're in a new realm of treatments now where we're actually hoping to intervene and either stop or reverse pathology within that, so slow people's decline down. We've got a huge amount of patient interaction in the space. There's more than 38,000 people taking part in clinical trials right now. That's a hugely important part of what we can do as a community to support the development of new medicines, and a lot of those are sponsored by industry. But across, I think I already mentioned the phase 1, phase 2, and phase 3 components of the therapeutics, and these are all the different mechanisms that are currently in those phases. So you can see a lot of the later stage assets are looking at amyloid pathology, so similar mechanism to lecanemab. Then we also have some in metabolism and energetics, symptomatic treatments that maybe restore cognition, and then various others that are listed in this. But I think what it shows you is that we're looking across a broad range of areas, and there's huge opportunity for the future. So now I'm going to hand over to my colleague, Paul Brennan from Oxford, who it's such a pleasure to work with and collaborate with on looking for new diseases, and I hope you enjoy his presentation. Thank you for listening.
Hi, everyone. Glad to see so many of you joining today. I'm going to try to share my screen here. Oh, I have to wait for Fiona to finish. While waiting for that, I'll just introduce myself. So, as Fiona said, I'm a chemist by background. I'm from the US, as you can probably tell from my accent, though I've lived nearly half my life in the UK now. And I've been working with Alzheimer's Research UK in drug discovery for the past 8 years. And a bit like Fiona, I was personally motivated to do this. I worked in lots of different areas over my career in drug discovery. I really wanted to do something that I thought would be the most impactful in terms of having the biggest, benefit to society. And Alzheimer's, I thought, was the one I could do that in. And secondarily, like many of you, I've personally been affected by Alzheimer's. I haven't had it myself yet, luckily, but loved ones have had it and have it as well, and live with it. So, and actually have passed away from it. So, I'm personally motivated as well. But I want to tell you a story about one of our drug discovery projects. I'm assuming you can all hear me okay. And we get to go back to the beginning and think about brain cells, because obviously Alzheimer's is primarily a disease of the brain. When we think about brain cells, the famous brain cells are neurons. In this box here, neurons are where all the thinking and feeling and seeing that we associate with the brain, sort of come from. But they're not the only cells in the brain. And in fact, neurons only make up about 40% of the brain cells, or the mass of the brain, the so-called gray matter. And it's the neurons that actually die when you have Alzheimer's disease, and lead to the decrease in cognition. But the other 60% of brain cells are what are called white matter, and there's a bunch of other cell types shown here. But the ones I want to focus on are called the microglia. And for a long time, people didn't really know what microglia were. And so this name just means small, non-neuron brain cell, essentially. But it's turned out that microglia are the immune system cells of the brain. So for a long time, people didn't even think the brain had an immune system, but it does. And the primary cells of the immune system of the brain are the microglia. And they're a little bit like macrophages, which you may have heard of. These are cells in the bloodstream, in the lymph nodes that prevent infection. And microglia are kind of like macrophages of the brain. So, why are these important? So this is an immune system cell, the microglia, and one of the big causes of human disease of all types is the immune system. So the immune system is this great thing that stops us from dying from microbes. Fights viruses, it fights bacteria. I think we all know a lot more about the immune system after COVID than we used to. But there's lots of well-known diseases, Crohn's disease, in the GI tract, neuropathy from diabetes that affects the nerves, fibromyalgia, lungs, psoriasis, arthritis in the bones and muscles, etc. All of these diseases, which are in very different parts of the body, are caused by the immune system essentially being overactive. And so the question kind of arose through a variety of strands of research that kind of started pointing to the immune system in the brain as being important in Alzheimer's disease. And so it was, this key question, is the immune system get overactive in the brain, just like it does in the rest of the body, to contribute or even cause Alzheimer's disease? And that's kind of where this project started. And you can think about the immune system in the brain as a bit like the gardener. I like this analogy, the gardener analogy. So here's everybody's favorite British gardener, Monty Don. And, Monty can do great things for the garden. He can trim out the dead leaves. He can pull out the weeds, and he can make the garden look great. But if Monty's having a bad day, he can also go too far, and he may start cutting branches that are healthy and happy. And so the immune system in the brain is like that. It's Monty essentially going too far and actually destroying healthy things, as well as getting rid of the bad stuff in the brain. And so in a bit more complexity, this is actually a key study that really highlighted why the immune system is important in the brain and important in Alzheimer's. So this is a complicated graph, but I'll walk you through it. And so these are all gene names, all these acronyms here. And so you may have seen some of these, like ApoE4, famous in Alzheimer's, amyloid precursor protein, also famous in Alzheimer's, but some that are less famous. And there are variants in these genes. We know we're all genetically different. We have different hair color, different eye color, we have different heights, different weights. And so, we all have variants of different genes. And in some cases, there are very rare variants in these three genes, the presenilin and APP genes, that are associated with Alzheimer's, and they are intimately associated with amyloid formation. And this is where we've really focused research efforts on Alzheimer's over the past 30 years, as Fiona said. And the two drugs which have been given accelerated approval recently, lecanemab and aducanumab, target essentially this system. And luckily, variations in these genes that cause early onset Alzheimer's are very rare. But if you have these variants, then you may get it in your 40s or 50s. But there's a lot of other genes shown here, these other circles, where they're actually much more frequent in the population. And so 50% of people have variations in some of these genes here, like INPP5D, that are associated with a risk of Alzheimer's, either slightly greater risk of getting it or slightly lower risk of getting it. And if you start to look in what these genes actually do, so I've circled some here, these ones in the blue boxes are all part of the immune system. So these are all genes expressed in these microglial cells in the brain, which are the gardeners of the brain, and variations in these genes. So slight differences between us actually change the risk of Alzheimer's disease. And so by looking at the immune system, we can actually try to modulate that. And these variations are pointing, sort of flagging that the immune system is important. So one of these genes that is important in the immune system is called the inflammasome. And as the name implies, it's to do with inflammation, specifically inflammation in the brain for Alzheimer's, although it's also active in the rest of the body. And the other name for this is NLRP3. And there was a lot of papers, showing the links between the inflammasome and Alzheimer's disease and other dementias as well, including frontotemporal dementia, as well as Parkinson's. So I won't go through these in detail, but there's a bunch of scientific papers highlighting the link between this gene and Alzheimer's disease. And so we wanted to develop an inhibitor of this inflammasome to use as a treatment that would change the immune system in the brain, and the hypothesis is, improve development of Alzheimer's, to stop it from happening. And so the best place to start a drug discovery project is what somebody else did. And so Pfizer had discovered back in 2000, the big drug company Pfizer, this compound called TRID3, and they showed that this was a potent inflammasome inhibitor. So this is a great place to start. Now, you can't use this drug directly in Alzheimer's treatment because, first of all, it doesn't even get into the brain. This stays in the bloodstream, in the tissue, but it doesn't actually penetrate the blood-brain barrier and get into the brain. And so Pfizer took this forward to develop for other inflammatory diseases. We saw all that list of them on my previous slide, but they showed actually this molecule was not safe enough. So it was too toxic to use in inflammatory disease. So what our hypothesis and what we have been doing is trying to make a better inflammasome targeting drug that will get into the brain and will be safer. And one of the ways that we do that is by taking the three-dimensional structure of the inflammasome protein itself and molecules like CRID3 that are bound to it, and try to design molecules that will inhibit it in the same way, but be more likely to get into the brain. And like all biology, I'm just a simple chemist, but all biology, when you start to study, is horrifically complicated. And so the scientists I work with have, and people all around the world have really built this picture of how the inflammasome gets activated, which I won't take you through, but how that causes neuroinflammation. And we are able to look at this in cells and study inflammasome activity and study what happens to it when we treat with inhibitors. So we have a deep understanding of the inflammasome biology in cells, and we are able to test molecules using cells engineered to look at this system. And as I said from the last slide, we have a 3D structure of the inflammasome to design new drugs. And then we will test those drugs in cells and also in mice. And there were some questions in the chat about animal studies, and so we can talk about that as well. But we will test these molecules in rodents as well as other species, and we definitely have to for safety. And obviously, so having a drug which gets into the brain and treats Alzheimer's is important, but it's not the only thing that determines whether a drug is ultimately successful. And so the reason I really like being a chemist, a medicinal chemist specifically, is that, you know, there's so many attributes a drug has to have. It has to get into cells, and it has to bind to their target protein selectively. It has to get into the CNS for things that like Alzheimer's or diseases of the brain. Has to be safe, you know, not causing cardiac effects. It has to last long enough to have an effect. It has to be stable to metabolism to last long enough. You know, usually these are pills, and not all drugs are pills, but, you know, things that taste bad tend to not to be good drugs. Has to, you know, get into the stomach and dissolve and be stable to the harsh conditions of the stomach, and then get absorbed into the body and ultimately excreted. So there's lots of things that go on in drug discovery and good drugs, as opposed to just being potent and selective. And ultimately, these things are products. You know, they have to sit on a shelf in the pharmacy and Boots, and be stable and importantly affordable as well. So I will finish there. I will pass on the baton now to Susan, who's gonna oh sorry, no, we're going back, sorry, we're not going back, we're going back to Fiona, who's going to take us through the questions and get us started in the discussion. So thank you very much.
Thanks so much, Paul.
Sorry, not Fiona, Josie, sorry.
That's all right. We're all joining back for the Q&A anyway. So I will just welcome Fiona back to the screen, and also Susan as well. So thanks Fiona and Paul for some really, really brilliant and really interesting talks. It's so fascinating to hear about your work at the Drug Discovery Institutes. So we have some pre-submitted questions, which some of you submitted before when you were signing up for the event today. So I thought I'd start with one first for Susan. So Susan, what are the most promising strands of research for treatment of Alzheimer's disease, do you think, coming up?
Let me make sure I unmute myself. Hi, thank you everybody for attending and for all of your questions so far. I'm Susan Kohlhaas. I'm the Director of Research and Partnerships at Alzheimer's Research UK. So if you haven't seen me before, that's who I am. And I really find this hard to pick one area to focus on. I think we're at a really exciting time in terms of understanding how to diagnose and detect diseases like Alzheimer's disease, and we're starting to see some movement in using things like blood tests to detect the proteins that build up in the brain. I think that's really exciting, but that's only really exciting if that helps us get treatments to people more quickly and more easily. And that's where I'm really excited. So Fiona said in her presentation, we've got, you know, well over 100 agents in clinical trials for Alzheimer's disease alone. And I think for me, there's probably going to need to be several different things that we're working on at the same time in order to really slow, stop, or reverse the diseases that cause Alzheimer's, or the things like Alzheimer's that cause dementia. And so I think we're making some great strides in terms of finding treatments for the proteins that build up in the brain. So many people have heard of a drug called lecanemab that can actually remove amyloid, which is one of the proteins that builds up in the brain as a hallmark of Alzheimer's disease. That's really exciting. I think there's a lot of work happening in that space, and we look forward to seeing progress in that space. But I think looking beyond that, we're also really interested as a funder of and as a charity interested in finding cures for diseases like Alzheimer's. How do we make sure that the brain is as resilient as possible to aging and damage as it happens? So how do we promote the cells in the brain to be as strong as possible and resistant to that damage? That's really interesting. That's what a lot of the work of the Drug Discovery Alliance is looking at. And then longer term, how do you actually start to restore function as well? And this is one of the areas where when I talk to people, they think, oh, that's a little bit of science fiction. But actually, when you look at the research that's happening, there are things that you can do to try and restore function and regain function. And that's an area of importance as well. And what I'm really looking forward to seeing over the next 5, 10, 15 years is progress in each of those 3 areas. And if we can do that, and if we can have several different treatments in each of those areas targeting different aspects of the diseases, then we can actually make real progress. But I'm really aware that I'm not the world's expert on this, so I'm really keen to hear from the experts. So I'm going to hand this back to Fiona and Paul really to answer what's really exciting for them as well.
Do you want me to go first, Fiona? I mean, as I said, I'm interested in neuroinflammation, and there's the first few drugs are being tested in neuroinflammation in Alzheimer's, and I think that's really exciting. And those drugs may also be used for other forms of dementia, which is one of the questions in the chat as well. Neuroinflammation seems to really be important in lots of different kinds of neurological diseases. FTD, Alzheimer's, Parkinson's, maybe vascular dementia as well, although I don't think the links are strong. And so seeing the first neuroinflammation drugs being tested in the clinic now, I think is really exciting.
Yeah, I think I'm like you, Susan. I'm like, how do you pick one in this field? You know, I think that the field is moving so fast right now that we're sort of on a wave of, you know, exciting changes. I think for me, one of the most exciting things, I think I touched on it in my talk about biomarkers and the way that we can see in living people what changes during the trajectory of disease. So right from before people show symptoms through to when they start to decline in various different functional realms, and then how that continues over the course of the disease, and how that's different from one individual to another. So we're starting to understand now that some people have resilience in their genetics that make them less susceptible to decline than others. We're starting to see that some people have more risk, some have some risk factors, some protective factors that interact with each other. So the molecular changes in terms of our understanding of the field are really allowing us in the drug discovery space to have a much bigger opportunity for intervention. And it's a big answer, but the second part of that is that everyone's dementia is different and every type of dementia is different. So being able to understand which components are driving the disease and the symptoms at different stages will allow us to maybe have a different way of treating people therapeutically, like we do in other diseases already, like cancer, where we treat based on different components with multiple therapeutics. So I think that we're still in the really early stages, but the more tools to treat people we have in our toolbox through lots of different therapeutic agents, the more we'll be able to do in the future to treat people at the right time with the right medicine.
Great, thanks so much, everyone. We had a question typed, Paul, I can just see you've typed the answer to that one, but maybe we'll ask so everyone can hear it as well. Why do the drug names so often end in mab? So lecanemab, aducanumab, gantenerumab, donanemab, they do all end in mab. Paul, could you answer that so we can all hear it as well?
Sorry, I didn't know it was
no worries.
So mab is short for monoclonal antibody, which a lot of the new drugs for dementia, as Josie said, but also lots of other diseases, that's the kind they are. I mean, there's different kinds of drugs, and the antibodies are the kind of been around for about 20 years and are the kind of, some of the best-selling drugs in the world. So they tend to end in mab.
Great, thank you. We had a question from an anonymous attendee. So they say that they appreciate the vast complexity of neurological disease. However, does progress and research such as this get shared with other research? So they're specifically interested in MSA, which is multiple system atrophy, I think. But perhaps maybe we could speak a bit about other neurological conditions as well, and how that is then enabling further understanding for other neurological advancements. Susan, perhaps I could come to you for that one first.
Yeah, I mean, I can start, and then again, Fiona and Paul, please do chip in. I mean, I think this is something that, we as a funder want to learn from other disease areas, and we want other disease areas to learn from what we're doing. So we are interested in funding research into all forms of dementia. So that's not necessarily, even though our name is Alzheimer's Research UK, it's not necessarily just Alzheimer's. We fund work on Parkinson's, which can cause things like Alzheimer's. We fund work on motor neuron disease. And I think what we're really interested in, and I think as a field, what we need to do is look at what the specific mechanisms are that are driving the disease and treat those. And there's huge crossover between those across many neurodegenerative diseases. So we can't be looking in our silos and thinking that we'll have the answer independently. And that's one of the real selling points of our Drug Discovery Alliance, is that we've tasked brilliant scientists from from all over the world to come together and identify what are the best pathways to look at, what are the best targets that we can look at, and then how do we get those through into industry as quickly as possible. And I think having that focus and not necessarily this is the area I'm working on and this is the target I'm working on and I'm not really interested in other things isn't really the approach we take as a funder. We're much more open than that, and we hope again that we, you know, the MAB, the monoclonal antibodies, they actually came and sort of started in the cancer field, and lots of disease areas have benefited from that. And again, I am hoping that in 20 or 30 years time, people will turn around and say, well, we really learned that from the dementia field, and that's what we hope to achieve as a funder.
Thank you, Susan. Paul and Fiona, anything to add to that?
The thing about sharing results is interesting. You know, we try to publish our results, and we probably don't do it as fast as we should. And there's a bunch of reasons for that. But I think, you know, that's a primary way we share results, and going to conferences and talking about things, and actually networking at conferences and talking to people from different fields is a great way of kind of cross-fertilizing ideas. I don't really know very much about MSA, but I kind of echo Susan's point that, you know, testing things, lots of drugs are used for not what they were first discovered for, and so we shouldn't be so arrogant to think that we always know everything. And so we should always be opportunistic and test things in other areas as well. And I think scientists are more open to that than they used to be.
Great, thank you both.
Yeah, I can maybe add to that as well, just quickly. One of the beauties of sitting in these clinical centres of excellence like UCL, Oxford, Cambridge, where we have access to clinical samples and experts in different neurodegenerative diseases, makes it really easy for us to share ideas and think through common mechanisms. So there is, I'm sure, underlying neurodegenerative pathways that are involved in why neurons die that are the same, and some that are different in the different diseases, which is why in MSA and Parkinson's you see basal ganglia degeneration in more so than the cortex, whereas in, you know, in Alzheimer's, the cortex and hippocampus, so different brain regions often die more predominantly in the early stages of those diseases. And that's the kind of thing we don't know for sure why that happens, but they're all parts of building the puzzle around all of neurodegenerative diseases. And I think, as Susan said, we are focused across diseases to make sure that we bring learnings from some of the clear neurodegenerative diseases that have more clear genetic drivers, for example, versus the ones that are much more complex with much broader contributions from different mechanisms that are causing the disease. So I think sometimes we can use diseases where there's a more straightforward pathway to understand. None of them are straightforward, but, you know, if they've got a gene that we know always causes the disease, we can use clues from that to understand the bigger diseases where there's more complexity. So I think it's definitely an area where we collaborate. I'm actually sitting right now in my hotel room in Brighton because I've been sharing science all week at the BNA, the British Neuroscience Festival of Neuroscience. So I think a lot of our work is sharing our ideas and discussing science with others, and, you know, that's one of the best parts of the collaborative nature we're working in at the DDA.
Amazing. Thanks so much, Fiona. So perhaps sort of following on from this discussion, but maybe more specific to dementia, Elizabeth asks, will future potential treatments for Alzheimer's disease be completely different for those for vascular dementia? So I think, Paul, you touched on that a little bit about how there might be some similarities across the two diseases and the two causes of dementia. Could you expand on that a little bit, maybe?
Yeah, I mean, I'm not an expert on vascular dementia. I mean, I have heard it said that, you know, they may even be the same disease, vascular dementia and Alzheimer's. Just one has, you know, the vascular dementia obviously has more vascular component with the blood supply, and then Alzheimer's has, you know, more of a non-vascular piece. But I suspect, and this is very much an opinion, that they will not be completely different, and that they will, you know, treatments will be tested in both. And the way to start doing that is to test, you know, treatments in the lab in models of Alzheimer's and models of vascular dementia, and see if we see the same things. Fiona's a neuroscientist. Maybe she has a better answer than me.
Well, I also have the benefit that I went to a talk this morning by one of the world leaders in vascular dementia at the conference, and I actually sat on a brainstorming panel with her about vascular dementia last week, so maybe I feel like I'm well versed to answer this question today more than there was 2 weeks ago. Which, but no, I mean, I think one of the things I think is very interesting about vascular dementia, and it doesn't make it unique, is that it's driven by small vessel disease, and so there's lots of different types of comorbidity that drive small vessel disease, all like diabetes, hypertension, but they're not necessarily guaranteed to cause it, but they are risk factors for small vessel disease, which then can translate into vascular dementia. So I think that it's one of the ones though where there is some interest in data to show that with appropriate treatments, you have a chance to reverse some of the components in early stages of the disease, because there's a lot of interest right now on fluorine endothelial cells that form the vascular connections within and line the blood vessels within the brain, and how they change in disease. So there's some new therapeutic areas emerging. We actually have a project at UCL that's looking at brain endothelial cell rescue and function to restore vascular health in disease. But I think that there will be some treatments that might cross both, but there might be different treatments. It depends, I think, on the disease.
Great, thank you both. Somebody has asked, and we've had a couple of questions sort of about time horizons and how long things might take. So somebody says, what is the probable time horizon for neuroinflammation therapy to be available? And also, what would be the mechanism for administration? Would it be an infusion therapy? Would anyone like to answer that one?
I can start.
Or parts of it, perhaps.
I'll have a stab, and then fill in the blanks and correct my mistakes. So there's 2 companies, Denali and Elector, who have TREM2 monoclonal antibodies in clinical trials. I think they're both in phase 1. So at the time horizon, I think it'll be 5 years minimum, probably longer. And the method of treatment will be they're treated IV, so intravenous injection.
Great. Thank you.
But Susan, is that about right? Or
yeah, I think the phase 1, I think that probably longer time horizon than 5 years. But I'd say, yeah, kind of 5 to 7 maybe is, and then, yeah, they're IV. But I think, again, with a lot of these treatments, what we would expect to see are also improvements in how they're administered. So the current treatments in clinical trials are infusion, but again, as we get some treatments for dementia, we will hopefully start to see things like injections that you could do at home, or tablets that you could take instead of an infusion, but we're not there yet. So we need to kind of wait and see, and I think that will be the next step once we start to see treatments becoming available.
Great, thank you, everyone. I think we've got time for perhaps 1 or 2 more questions before we wrap up for today. So would anybody like to answer the question, which is, what is currently the most promising therapy for targeting tau pathology?
I don't know, because we haven't got that many readouts for those tau trials. So I think I've got the luxury of not being a little bit dispassionate about it, because I haven't actually developed these things. I can kind of sit back and say, okay, what does the data show us? I think the most promising one will be the one that reports out positive clinical trials. So I think it's for us at Alzheimer's Research UK, it's a matter of, let's look at what's out there. Let's continually monitor for the results, but it's that phase 3 clinical trial result that we're really looking for in order to have confidence that this could go through to a regulatory approval stage, and that's what we're always looking at.
Great, thank you. I'm just going to have a quick look at some of the pre-submitted questions that we had come through as well, just to make sure we've sort of covered everything that people were asking.
Maybe we could just add to that tau question, just in terms of the modalities, because I think tau is interesting in that there's various different modalities in clinical development now to modify tau. So we have monoclonal antibodies like there is for amyloid, but there's also small molecules that target the aggregation of tau. I'm not sure exactly where they're up to in clinical trials, but I know there's various different small molecule therapeutics which would offer a potentially a pill-based approach. And there's also antisense oligonucleotides, which are antisense oligonucleotides are like little short nucleic acid sequences that are in the opposite sequence to the complementary in sequence. Sorry, that's a very bad way of describing it. But basically what they do is they cause the body to break down the tau as it's expressed and prevent the buildup of it. And that would hopefully stop the aggregations. And they are delivered to the spinal cord. And so there's three different, as far as I know, there's three different types of modality being developed in the clinic right now for tau. Which obviously is really interesting. And I think, but I agree with Susan, we're all excited to see what those therapeutics show in clinical trials, because, you know, we really need to show efficacy before we can confirm our thoughts on which one will be the best for patients.
Great, thank you, Fiona. And just as we've got 5 minutes left, I'll just ask you all one more question, which will be the same question for everyone. So perhaps Paul, will come to you first. So what would you say is your biggest hope for the future of dementia research in general?
I'm excited by people looking beyond amyloid. I think we've got 2 drugs that unequivocally answer whether amyloid is important, and people looking at other mechanisms. We talk about them, tau, neuroinflammation, other things. So that's really exciting, and we're going to see results from these, and I just hope they're positive. You know, the accelerated approval of 2 drugs in the last couple of years is a huge breakthrough. We haven't had any new disease-modifying drugs for you know, ever, and so this is a massive thing. And I think the ball is getting rolling now, and we're going to see some treatments. I don't know if there's a question about cure. I don't know if we're going to see a cure, but I think we're going to have treatments that will stop or really slow down the disease.
Amazing, thank you. Fiona, same question. What's your biggest hope for the future of dementia research?
Ooh, I think when we have a toolbox to identify before symptoms and stop the decline, so that patients can have, you know, people with risk of dementia will no longer develop it, and then, you know, families will be able to live without the terrible experience of losing a loved one in this way. So definitely, I think the field is moving towards that in terms of understanding the mechanisms that drive both the early symptomatic stages of the disease, but I think we've got to go another step further to pre-symptomatic stages of the disease to really cut it dead before it starts. So I'm excited to see that in the future.
Amazing, thank you. And finally, Susan, same question.
Yeah, so I'm really excited to see the advent of the first generation of treatments that can kind of slow disease progression. I think if you look at other fields, so I previously, prior to this, worked in the field of multiple sclerosis. We didn't have anything for 150 years. We got our first disease modifying treatment that could, you know, reduce the number of relapses that people had. And then you just see the pace of change after that point in the field, and how quickly we had to redesign our services and how people were treated, but how that fundamentally shifted how quickly we could do research and make more progress. So we went from, you know, 150 years of describing the symptoms of MS to having our first treatment, to having, you know, by the time I left that field and came to dementia, we had almost 20 drugs available on the NHS, and that was in a time horizon of kind of 25 years. And that's what I hope that we see this first generation of drugs doing for dementia. It makes people think about, okay, well, maybe there is something we can do about this disease, and maybe I should go and see my doctor coming forward for a diagnosis. It makes us think about, oh, wait a minute, how do we diagnose people as quickly as possible and get them into the system? And then how do we use that that energy to try and research more and get more treatments, more better treatments as well? This is just really the start. There's no area that has usually cracked a problem with the first generation of treatments. It's building on that, and what we do with this now, it's going to be essential.
Amazing. Thank you so much, everyone. So just as we approach 5 o'clock, I'd like to round off the event today by thanking Fiona, Paul and Susan for joining us and answering all of our questions. It was really brilliant and really amazing talks and really fascinating to hear all about your work. So thank you all so much for joining us.
Thank you. It was great to be here.
Yeah, thank you for having us. It's a pleasure to talk about our work.
Thanks for attending. It's great to see so many people.
Yeah, thanks everyone. So just before you go, we've got a few poll questions that we'd love you to answer, just to give us a quick insight into how you found today. So those will be appearing on your screen now, and we will be sending out a more detailed, a link to a more detailed feedback survey with the video recording and subtitles next week. So please do complete the survey as well, and we'd appreciate any feedback you can give to us about the event. So the polls that should be on your screen now, were the contents of the talks too technical, about right, or too simple? Would you recommend Lab Notes events to your family and friends? And do you think you've learned something new about dementia research at the event today? So I'll just give a couple more seconds for you to answer those questions. And then we will pop those on the screen of the answers. So fab that most people have said that the event was about right today, and lots of people would recommend Lab Notes, and lots of people have learned something new, which is brilliant. I know that there were a few questions that we weren't able to answer today, and some where you might need some more personalised information or guidance on where to turn. So our dementia research info line is there to help. Hopefully on the screen now you should be able to see the phone number and email address for the info line. So they can answer questions and also signpost to other sources of information and support. So please do get in touch with them if you have any more questions. You may also be interested in finding out more about joint dementia research, which can match you to clinical research studies taking place. This is open to anyone over the age of 18, and you do not need a dementia diagnosis to take part in the studies. One study for using lecanemab in people at high risk of dementia is still recruiting through JDR until the end of August. So you can find out more about getting involved in this, and if you might be a potential match or possible candidate on the JDR website, the Join Dementia Research website. So have a look on there. We would love to hear what you thought about today's event in more detail, so please do fill in the survey and let us know your thoughts. You should be able to scan this QR code on your screen, and there'll also be a link in the chat and also in an email after the event. If you've missed any of our previous Lab Notes events, you can watch them back in your own time, as they are all available on the Lab Notes webpage or over on our YouTube channel. We've covered topics like EDEN, which is our early detection of neurodegenerative diseases. We did that one back in the summer. We've talked a bit about detection today. We've covered vascular dementia. We've covered things like sleep and dementia. So loads of really, really interesting topics from our previous Lab Notes. So do check them out. And finally, I want to say a huge thank you for joining us today. I hope you found it interesting and useful, and we hope to see you again soon. Thanks, everyone.